This is my book on protein synthesis. There's some basic vocab you would need to know is mRNA, which stands for messenger RNA, and it helps take genetic info to the ribosomes. The second word you would need to know is tRNA, which stands for transfer RNA, and it helps connect the mRNA and the amino acids together. And then the third word would be amino acids, and it's a protein that makes up a lot of things in our body. And then the fourth word would be ribosome, a particle made of RNA that binds the mRNA and tRNA into protein synthesis. So to get some background information, DNA. So DNA stands for deoxyribonucleic acid. It's in every living organism, and it carries the organism's genetic information. It duplicates itself when cells are going through mitosis, and it's a double helix as shown here. A double helix consists of two chains that coil around each other, and they're attached by nitrogenous bases, which is represented by these ladders here. And the four nitrogenous bases are thymine, adenine, guanine, and cytosine. So to get some background on DNA structure, so DNA is a double helix because there are two strands of a sugar phosphate backbone, which is this gray part, and they go opposite directions. So one goes up this way, and then one goes down this way. Connecting the two strands are your nitrogenous bases, which again are represented by this ladder. And there are four bases, again, which are guanine, cytosine, adenine, and thymine. However, only certain bases can go together, so adenine and thymine go together, and then guanine and cytosine go together, but they can't go with anything else. So the bases are connected and held together by hydrogen bonds, and DNA is just made as a whole, it's just made out of nucleotides, and nucleotides are made of a phosphate group and a sugar, which is what is found in the backbone, and then a nitrogenous base, which is the latter part. And so then the reason why it coils around each other is because of the angle of the nitrogenous base bonds, and then the way the ingredients of the backbone. Okay, so replication. Replication is when a DNA double helix is being copied to make two identical DNA molecules. So during this process, each strand is like a template, so it could be copied exactly. So they'll split out, there'll be one strand of DNA, it'll split out, and then it will match these together with another, with their proper nitrogenous base matches, and then we'll create two new strands of DNA. Okay, so RNA. RNA is a biomolecule, and it's like a copy / template of DNA. It carries the same genetic information as the DNA. It is not used for long-term storage. Its sugar is ribose rather than deoxyribose. So in DNA, the sugar is deoxyribose, which is why it's DNA, but in RNA it's just ribose. And then it's only single-stranded, so as you see here, instead of there being another coil going around connecting to it, there's only one strand. So it's kind of like half of the DNA. And then it doesn't have the base thymine, it actually has the base uracil, which takes its place. So A goes, if you were to look at a diagram, the letter A would be matched with the letter U rather than the letter T. Okay, so transcription. During transcription, DNA is converted into mRNA. mRNA is messenger RNA, that's what it stands for. Transcription occurs in the nucleus, and the promoter sequence tells the polymerase where to start. In transcription, the four bases are adenine, guanine, cytosine, and uracil, so again, there's no thymine. And when transcription is taking place, the genetic code is grouped into threes. These are called codons, which are shown up here. Polymerase synthesize RNA, and it's an enzyme that helps copy the DNA sequence into an RNA sequence. So again, up here's a codon example. So this right here is a G, so its match would be C. This is a U, so it'd be A, and then the A is a U. As you can see, those two would fit together like a puzzle piece. Okay, translation. So this is my Google Translate box. Translation is when proteins are made using the copy of the DNA on the mRNA. So transcribed mRNA directs the translation process, and to start translation, tRNA molecules bind to codons that carry amino acids. Ribosomes help form peptide bonds. First, it breaks the bond holding the first tRNA molecule to its amino acid. Then new ones come in to replace it. In the process of translation, there is tRNA, rRNA, and mRNA. So here, this is what it was talking about over here. So this would be fitting here, but then new one comes in, and this one gets knocked out just to keep replacing. And so here it says translation happens in the cytoplasm of cells and in the ribosomes, and here proteins are made of amino acids that link together to form a polypeptide chain. Then these chains fold into a 3D structure. So there gets to be so many of them that they kind of come together and create a 3D structure. tRNA contains of two main parts, a large subunit and the small one. The structure is an L shape, so it's kind of shown here, and it bonds to amino acids, and the other side has an anticodon. So this is the amino acids here. Okay, so ribosome to protein, how does this happen? So it's kind of like a summary, so overview. First, the DNA is connected by a nitrogenous base or genetic code. Due to this, transcription occurs and copies the genetic code from DNA to mRNA. Next, the mRNA has all of the info, so translation happens. During translation, the mRNA info creates proteins by creating polypeptide chains. And then finally, for the physical protein to be created, the polypeptide chains fold over one another and create a 3D figure, or kind of an object, which is considered a protein. So this is a protein structure. Here, they are binded together by peptide bonds. Okay, this is just some extra info that you would want to know that has to do with protein synthesis. So how does the ribosome know when to start and stop translating the mRNA? So there are actually start and stop codons. One of the stop codons is UGA or UGA, as we like to say in class, and so it'll tell the ribosome when to start and stop. And then what are the peptide bonds and where they found in proteins? A peptide bond is a chemical bond that links amino acids together, and proteins that are found between amino acids. So if you have two amino acids, they're held together. If these are two amino acids, in the middle here would be a peptide bond holding them together. So the importance of all of this information, this is all important because without this information, we wouldn't know how our body works. We also would not be able to do certain experiments in science, and also helps us understand and cure diseases that are related to our proteins and our DNA. And then finally, it helps us understand mutations. So if we were to have a sickness or something was going on in our DNA, we actually know what the DNA structure looks like and how to deal with it and the different parts of it, which makes it 10 times easier to go through and be able to solve the problem. Okay, and then last but not least are mutations. So this is all about mutations. Mutations are changes / errors in genetic information that can be passed down to offspring by parents. So it can be inherited from the parents by the offspring. So there are two types of mutations, gene mutation and chromosomal mutation. Chromosomal mutation is typically more severe, but because it's changing a whole chromosome rather than just one gene. So these can either be beneficial or neutral, or actually impact the organism negatively depending on their environment and what they need to survive. So real life examples would be an orange tiger to a white tiger, long beak to a short beak, so in different birds depending on we learned about Charles Darwin last year and the different birds that he studied and the different types of food they ate. Their beak just kind of adapted to it on its own because of mutations. So then if you don't have sickle cell anemia, you can actually develop it. And then here, so this is an example of a mutation. So here it goes red, blue, green, yellow, red, blue. Here it's red, blue, yellow, which is actually different from it being over here. So that would be a mutation. And that is all. Thank you.